Good morning all. Today we'll discuss about meiosis. You know about mitosis, which is an equational division where daughter cells receives the same number of chromosomes as that of parental cell. Daughter cells ಗಳು parent cell ನಷ್ಟೇ chromosome ಅನ್ನು receive ಮಾಡ್ತದೆ mitosis ಅಲ್ಲಿ. Meiosis, which is a special type of cell division that takes place in the case of sexual reproduction. The offsprings which are produced in a sexually reproducing type where gamete formation takes place, and formation of gametes, that process is called gametogenesis. This gametogenesis process of formation of gametes, that is by the help of meiosis. Offsprings produced by sexually reproducing organisms where there is a process called gametogenesis. In gametogenesis, there is a division called meiosis. This meiosis which results in the formation of haploid gametes. Haploid gametes with a meiosis and fertilization which results in a restorage of original diploid set of chromosome in a diploid organism. Organism diploid ಆಗಿ ಅದು sexually reproduce ಮಾಡ್ತಾ ಉಂಟು ಅಂತಾದ್ರೆ ಅಲ್ಲಿ haploid gametes ಬರ್ತದೆ. ಆ haploid gametes ಬರುವುದು gametogenesis ಹೇಳುವಂತ ಒಂದು ಪ್ರೋಸೆಸ್ ಅಲ್ಲಿ. Gametogenesis ಅಲ್ಲಿ meiosis ಆಗುತ್ತೆ. So these gametes which are haploid in nature, these are produced by the gamete mother cells, or these specialized cells which are called gamete mother cells, which are diploid in nature, and meiosis takes place in that cell. Gamete mother cells which undergoes meiosis. Okay, gamete mother cells undergoes meiosis and produces haploid gametes. And this meiosis which ensures the haploid phase of the sexually reproducing organism in a diploid body. And after the meiosis, we got the haploid gamete. Once again, there is a fertilization, fusion of two haploid gametes is there. Gametes will be there, right? And it results in a restorage of original diploid cell. So, meiosis alli diploid organism, right? And here there is a meiosis which results in the formation of haploid gametes. Two sets of chromosomes in a diploid body, and in a gamete mother cell, these are the specialized cells where meiosis takes place, right? And that after the meiosis, formation of haploid gametes will be there, right? Therefore, meiosis which ensures a haploid phase of the sexually reproducing organism. And after the gamete formation, say another body also where there is a result of haploid gamete, fusion of two haploid gametes, that is nothing but fertilization. This process which is called fertilization, fusion of two gametes results in a diploid set. That means restorage of original diploid cell or original chromosome number. Okay, original chromosome number. Meiosis alli sexually reproducing organism alli haploid gametes ಬರಬೇಕು. ಅಂದ್ರೆ ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ haploidic ಲೈಫ್ ಸೈಕಲ್ ಉಂಟು, ಡಿಪ್ಲಾಯ್ಡ್ ಉಂಟು. ಆದ್ರೆ ಮೇಜರ್ ಪಾರ್ಟ್ ತಗೊಂಡ್ರೆ ಡಿಪ್ಲಾಯ್ಡ್ ಅಲ್ವಾ? ಹಾಗಾಗಿ haploid phase ಹೇಗೆ ಸಿಕ್ಕುತ್ತೆ? Haploid phase where that is because of meiosis. Haploid phase results, that is because of meiosis. Fertilization, it results in a diploid condition, restorage of original chromosome number. That original chromosome number, it is maintained in this fertilization. Meiosis ಅಲ್ಲಿ haploid ಆಗುತ್ತೆ, fertilization ಅಲ್ಲಿ haploid gamete ಗಳು ಫ್ಯೂಸ್ ಆಗಿ diploid condition ವಾಪಸ್ ಬರುತ್ತೆ. This is about the meiosis. Okay, it takes place in in the case of animals, in plants also, fertilization. Gametogenesis is a process where haploid gametes are produced by the diploid cells, specialized cells. ಯಾಕೆ ಎಲ್ಲಾ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಮಿಯೋಸಿಸ್ ಆಗೋದಿಲ್ಲ. ಯಾವ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಗ್ಯಾಮೆಟ್ಸ್ ಬರಬೇಕು, ಯಾವ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಗ್ಯಾಮೆಟ್ಸ್ ಬರುತ್ತೆ, ಆ ಸೆಲ್ಸ್ ಅನ್ನು ನಾವು ಸ್ಪೆಷಲೈಸ್ಡ್ ಗ್ಯಾಮೆಟ್ ಮದರ್ ಸೆಲ್ಸ್ ವಿಚ್ ಅಂಡರ್ಗೋಸ್ ಮಿಯೋಸಿಸ್ ಅಂತ ಕರೀತೇವೆ. ಆ ಸೆಲ್ಸ್ ಗಳು ಮಿಯೋಸಿಸ್ ಇಂದ ಕೊಟ್ಟ ಗ್ಯಾಮೆಟ್ ವಾಪಸ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಟೈಮ್ ಅಲ್ಲಿ ಫ್ಯೂಸ್ ಆಗುತ್ತೆ. And once again there is a restorage of diploid original chromosome number. Okay, this is about the meiosis. And what it all takes place? What are the key points of meiosis? In meiosis there are two stages, two sequential cycle of nuclear division and cell division. That is, there are two times, two stages, meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. So first point, there are two sequential cycles of nuclear division and cell division, that is meiosis 1 and meiosis 2. But remember, there is only one time where DNA replication takes place. Only one time, one cycle of nuclear division, but two times that nuclear division and cytoplasm division takes place. That is in the case of meiosis. Next point, meiosis begins only after the duplication of DNA, which takes place in S of interphase. S phase of interphase. Andre meiosis yavaga start agutte anta helidre, gamete mother cell alli, in gamete mother cell, when there is a completion of DNA replication which takes place in a S phase of interphase, then only initiation of meiosis takes place. Okay, that is only after the completion of replication of DNA. Okay, replication of DNA which takes place in S phase of interphase. Okay, now meiosis, here one more special feature where homologous pairing takes place. Homologous pairing of chromosomes. What are these homologous chromosomes? I'll explain now. Pairing of homologous chromosome and recombination between them. Homologous chromosomes pair ಆಗುತ್ತೆ, ಅದರ ಮಿಡಲ್ ಅಲ್ಲಿ recombination ಆಗುತ್ತೆ, exchange of genetic material between these two that takes place, which is very important. And because of this recombination only, there is a genetic variation occurs which may end up with a speciation of, it results in a evolution. Meiosis ಅಲ್ಲಿ ಇದು ಮೇನ್ ಫೀಚರ್. ಹೋಮೋಲೋಗಸ್ ಕ್ರೋಮೋಸೋಮ್ಗಳು ಪೇರ್ ಆಗ್ತವೆ, ಆ ಹೋಮೋಲೋಗಸ್ ಕ್ರೋಮೋಸೋಮ್ ನ ಮಿಡಲ್ ಅಲ್ಲಿ ರೀಕಾಂಬಿನೇಷನ್ ಆಗುತ್ತೆ. ರೀಕಾಂಬಿನೇಷನ್ ಆಗುವಾಗ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಜೀನ್ಸ್ ಎಕ್ಸ್ಚೇಂಜ್ ಆಗಿ ನೆಕ್ಸ್ಟ್ ಗ್ಯಾಮೆಟಿಗೆ ಬರುವಾಗ ವಿತ್ ನ್ಯೂ ಕಾಂಬಿನೇಷನ್ ದಟ್ ಜೀನ್ಸ್ ವಿಚ್ ವಿಲ್ ಎಂಟರ್ ದ ಗ್ಯಾಮೆಟ್. ಹಾಗಾಗಿ ಗ್ಯಾಮೆಟ್ ಅಲ್ಲಿ ವೆರೈಟೀಸ್ ಆಫ್ ಜೀನ್ಸ್ ಇರುತ್ತೆ, ಯಾಕೆಂದ್ರೆ ಎಕ್ಸ್ಚೇಂಜ್ ಕ್ರೊಮ್ಯಾಟಿನ್ ಮೆಟೀರಿಯಲ್ ಇರುತ್ತೆ. That results in a recombination or which end up with a variation ಅಂತ ಹೇಳ್ತೇವೆ. That is homologous pairing. Homologous pairing takes place and there is a recombination also. Recombination. So meiosis 1 and meiosis 2, and here you can see another point where genetic only after the duplication of DNA, which takes place in S, then there is a initiation of meiosis takes place. And another point is homologous pairing of chromosomes and recombination between them. Another main point feature of that, meiosis, which results in 4 haploid cells. 4 haploid cells which are referred as gametes. Okay. Difference between mitosis and meiosis we'll discuss at the end. Now, about the meiosis. Meiosis, here are 2 sequential cycles of nuclear division and cell division. That is meiosis 1 and meiosis 2. But there is only one time DNA replication takes place. And another point is, only after the completion of DNA replication, there is an initiation of meiosis takes place. And main feature is a pairing of homologous chromosomes, recombination between those two, and another thing is 4 haploid gametes, 4 haploid cells are produced at the end of meiosis. Those are called haploid gametes. Okay, we'll summarize this. Meiosis. These are the key points of meiosis. As we said now, meiosis which involves 2 sequential cycles of nuclear cell division, that is meiosis 1 and meiosis 2. Okay, but there is only a single cycle of DNA replication. DNA replicates only once, but there are 2 stages, meiosis 1 and meiosis 2, where nucleus division and cell division that takes place. Second point is meiosis 1 that initiates only after the parental chromosome that have replicated. When there is a replication of DNA, here when mention the chromosome, parental chromosome replicate ಆಯ್ತು ಅಂತ ಹೇಳಿದ್ರೆ ಇದು chromosome ಅಲ್ಲಿರುವಂತ DNA replicate ಆಯ್ತು. Not about the multiplication of chromosome. Chromosome number which is not doubled. Okay, only replication of DNA, thereby chromosome where 2 sister chromatids are formed, right? That is to produce identical sister chromatids. ಒಂದೇ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿರುವಂತ 2 ಕ್ರೋಮ್ಯಾಟಿಡ್ ಗಳನ್ನು ನಾವು ಸಿಸ್ಟರ್ ಕ್ರೋಮ್ಯಾಟಿಡ್ ಅಂತ ಹೇಳ್ತೇವೆ. So in one chromosome 2 chromatids, right? Because duplication of DNA, chromatin material with DNA along with the protein, right? This DNA along with the protein that doubles, therefore we'll get 2 chromatids, sister chromatids. These 2 chromatids belong to this chromosome, right? Therefore these are called identical. This will be identical because our DNA replicate ಆಗಿರೋದಲ್ವಾ. 2 identical sister chromatids at the S phase. S phase ಅಲ್ಲಿ DNA replication ಆದ್ಮೇಲೆ meiosis first start ಆಗುತ್ತೆ. And main thing, that is, it involves pairing of homologous chromosomes. Pair of homologous chromosomes. See, in each diploid organism, in each cell, right? Each cell which receives one set from the father, one from the mother, right? After the fusion of 2 haploid gametes, that results in a zygote, where there is a mitosis takes place, cleavage, that is nothing but mitosis. It results in a multicellular form. In all cells, 23 pairs of chromosome that is present. Are 23 pair only, 23 from father, 23 from mother. In the form of gamete, it will come and fused, that is to form zygote. Zygote, that single cell that divides to give this multicellular body. Alva? ಎರಡು haploid set gamete ಅಲ್ಲಿ 23 ಇರುತ್ತೆ. ಈಗ 23 first chromosome, second chromosome, third chromosome, fourth, like that 23 chromosomes in one gamete, and first, second, third, like that from another gamete. After that fusion, zygote ಅಲ್ಲಿ pair 23 pairs. ಇಲ್ಲಿ meiosis ಅಲ್ಲಿ ಸ್ಪೆಷಲ್ ಆಗಿ ಏನಾಗುತ್ತೆ, ಕ್ರೋಮೋಸೋಮ್ ಗಳೆಲ್ಲವೂ ನ್ಯೂಕ್ಲಿಯಸ್ ನ ಒಳಗೆ ಫ್ರೀ ಆಗಿರುತ್ತೆ. ಆದ್ರೆ meiosis ಸ್ಟಾರ್ಟ್ ಆಗುವಾಗ ಪೇರ್ ಆಗಲಿಕ್ಕೆ ಶುರು ಆಗುತ್ತೆ. ಫಸ್ಟ್ ಕ್ರೋಮೋಸೋಮ್ ಫ್ರಮ್ ಫಾದರ್, ಫಸ್ಟ್ ಕ್ರೋಮೋಸೋಮ್ ಫ್ರಮ್ ಮದರ್. ಆ ಗ್ಯಾಮೆಟ್ ಅಲ್ಲಿ ಬಂದಿರುವಂತದ್ದು, there is a pairing of these chromosomes takes place. These are called homologous. Morphologically, genetically similar. ಆ 2 ಕ್ರೋಮೋಸೋಮ್ಗಳಲ್ಲಿ ಜೀನ್ಗಳು, ನಂಬರ್ ಆಫ್ ಜೀನ್ಸ್ ಇರಬಹುದು, ಯಾವ ಜೀನ್ ಇರುತ್ತೆ ಎಲ್ಲವೂ ಸೇಮ್ ಇರುತ್ತೆ, ಅದರ ಸೈಜ್ ಇಂದ ಹಿಡಿದು ಎಲ್ಲವೂ ಸೇಮ್ ಇರುತ್ತೆ. ಸೊ ದೋಸ್ ಆರ್ ರೆಫರ್ಡ್ ಆಸ್ homologous chromosomes. pairing of homologous chromosomes takes place, and between them there is another process called recombination. Paired chromosome ಅಲ್ಲಿ gene exchange ಆಗುತ್ತೆ. ಕೆಲವೊಂದು gene ಇಲ್ಲಿ ಡಾಮಿನೇಟ್ ಆಗಿರಬಹುದು, ಅದರ allele ಈ ಕಡೆ ಡಾಮಿನೇಟ್ ಆಗಿರಬಹುದು. See, that is, it may be in a recessive condition, not the dominant. In one chromosome it may be in a dominant nature, in another chromosome it may be in a recessive nature, right? Exchange of genes takes place between those homologous chromosomes. That is called recombination. Recombination, that is a main feature of meiosis. And 4 haploid cells, which are called gametes, will be produced at the end of meiosis 2. Okay, this is about the key points of meiosis. And meiosis substages that we know, meiosis 1 and meiosis 2. Meiosis 1 ಅಲ್ಲಿ ಏನೆಲ್ಲ ಇರುತ್ತೆ? Meiosis 1 where prophase 1 is there, prophase 1, metaphase 1, anaphase 1, telophase 1. Same thing in the case of meiosis second, it is also divided into meiosis second, where we can see prophase second, metaphase second, anaphase second, telophase second. These are the substages of meiosis. So, ಇದು ಮೈಟೋಸಿಸ್ ಮತ್ತು ಮಿಯೋಸಿಸ್ ಗೆ ಇರುವ ಡಿಫರೆನ್ಸ್. ಈಗ ಡಿಪ್ಲಾಯ್ಡ್ ಸೆಲ್ ಇಂದ ಹೆಪ್ಲಾಯ್ಡ್ ಬರಬೇಕಾದ್ರೆ ಮಿಯೋಸಿಸ್ ಬೇಕು. ಆ ಎರಡು ಹೆಪ್ಲಾಯ್ಡ್ ಸೆಲ್ಗಳು ಫ್ಯೂಸ್ ಆಗೋದು ಫರ್ಟಿಲೈಸೇಷನ್. ಫರ್ಟಿಲೈಸೇಷನ್ ಆಯ್ತು ಅಂತ ಆದ್ರೆ ಒರಿಜಿನಲ್ ಕ್ರೋಮೋಸೋಮ್ ನಂಬರೇ ವಾಪಸ್ ರೀಸ್ಟೋರ್ ಆಯ್ತು, ಝೈಗೋಟ್ ಅಂತ ಹೇಳ್ತೇವೆ. ಯಾವಾಗ ಎರಡು ಹೆಪ್ಲಾಯ್ಡ್ ಗ್ಯಾಮೆಟ್ಗಳು ಫ್ಯೂಸ್ ಆಯ್ತು, ಡಿಪ್ಲಾಯ್ಡ್ ಸೆಲ್ ಅನ್ನು ಗ್ಯಾಮೆಟ್ ಅಂತ ಹೇಳ್ತೇವೆ. ಆ ಡಿಪ್ಲಾಯ್ಡ್ ಸೆಲ್ ಇಂದ ವಾಪಸ್ ನಮಗೆ ಮಲ್ಟಿ ಸೆಲ್ಯುಲಾರ್ ಬಾಡಿ ಬೇಕು ಅಂತ ಆದ್ರೆ ಇಲ್ಲಿ ಮೈಟೋಸಿಸ್ ಆಗಬೇಕು. ಬಿಕಾಸ್ ಮೈಟೋಸಿಸ್ which divides and redivides, it increases the number of cells, multi cellular nature. ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರುವಂತಹ ಸೆಲ್ಸ್ ಟಿಶ್ಯೂ ಲೆವೆಲ್ ತಗೊಳ್ಬಹುದು, organ system grade of organization, it may be any one of that, right? ಅದು ಮೈಟೋಸಿಸ್. ಇಲ್ಲಿ ಮಿಯೋಸಿಸ್ ಅಲ್ಲಿ ಏನು, ಇಲ್ಲಿ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಗ್ಯಾಮೆಟ್ಸ್ ಬರುವಂತದ್ದು, ಡಿಪ್ಲಾಯ್ಡ್ ಇಂದ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಬರುತ್ತೆ. ಕ್ರೋಮೋಸೋಮ್ exact half chromosomes will receive, that gametes will receive exact half number of that parent cell. Parent cell ಅಲ್ಲಿ ಎಷ್ಟು ಕ್ರೋಮೋಸೋಮ್ ಇತ್ತು ಅದರ half set daughter cells ಗೆ ಬರುತ್ತೆ. 4 daughter cells are produced. Those 4 cells which are called gametes. Okay. Now one by one we'll discuss meiosis first, with the prophase first, metaphase first, anaphase first, telophase first. Now we'll study about the prophase first of meiosis first. Meiosis first with the substages, that is prophase first, metaphase first, anaphase first and telophase first. Now about the prophase first. Prophase first, which is lengthiest process as well as it is a complex process, and prophase first, which is subdivided into following stages, that is leptoteme, zygote, packetine, diplotene and diakinesis. So these are the sub-stages which comes under prophase first. Meiosis first ಅಲ್ಲಿ ಮತ್ತೆ sub-stages ಉಂಟು ಅಂತ ಹೇಳಿದಲ್ಲ, ಆ prophase first ಅಲ್ಲಿ ಯಾವುದೆಲ್ಲ ಉಂಟು? Prophase first, it is not simple as that of mitosis. It is much complex and it is the lengthiest process. And here, prophase first, once again sub-stages are there: leptotene, zygote, packetine, diplotene and diakinesis. In a first stage, that is in a leptotin stage, chromosomes starts to visible. Under microscope, we can see those chromosomes start to visible, and the chromatin fibers which are already duplicated, right? That takes place in the S phase itself. Duplicated chromatin fibers. Now that it is visible under microscope, and we can see the beaded appearance in that chromatin fibers, which are called chromomeres. If this is considered as a cell and within a nuclear envelope, here you can see the chromomeres, chromatin fibers which will be like this, and with a already doubled chromatin fiber, right? So this is about the chromomeres, that is in the case of leptotin stage. Leptotin stage, another chromosome with the same, that is chromomeres, beaded appearance, DNA which is multiplied, replicated already. So we can get two chromatids, right? This is in the case of leptotin. Chromosome already andre S phase alli DNA double ಮಾಡ್ಕೊಂಡಿದ್ದೇವಲ್ಲ, ಹಾಗಾಗಿ ನಮಗೆ ಲೈಟ್ ಆಗಿರುತ್ತೆ. Still it condenses. Condensation continues. Chromosome condense ಆಗ್ತಾ ಉಂಟು. ತುಂಬಾ thick ಆಗಿಲ್ಲ, thin thread like structure, but already in a condensing manner, right? So ಈ ತರ ಇರುತ್ತೆ. This is about the leptotin stage. Leptotin stage, chromatin fiber will start to visible, and condensation of chromatin fiber, chromosomes continues. This is about the leptotin. Next stage is zygote. Zygote is very important. In a zygote stage, there is a pairing of homologous chromosomes takes place. Pairing of homologous chromosomes. This is about leptotin. Now about the zygote. Say leptotin. ಆಗ ಬರೆದಿರುವಂತ ಡಯಾಗ್ರಾಮ್ ಅಲ್ಲಿ ನೋಡಿದಿರಿ, chromosomes which will be thin thread like structure only, which starts to visible, where we can see beaded appearance, that is called those are called chromomeres. Beaded appearance, okay, beads in a bead anta irtave, chromomeres, right? Now in a zygote stage, that homologous pairing takes place. Pairing of homologous chromosomes. Pairing of homologous chromosomes. Pairing of homologous chromosomes, and that is to call synapses. Synapses. Formation of synapses takes place. What are those homologous chromosomes? Say, in a diploid cell, that takes place in a meiosis, right? In a diploid cell, one chromosome from father which we receive, right? And its partner chromosome from mother will receive, right? Those chromosomes which will be free in a nucleoplasm, there is a pairing takes place in a zygote stage. Homologous chromosomes pairs. Pairing of these homologous chromosomes, that is called synapses. Now these pair, that is called bivalents. Bivalents. Why these are called bivalents? In one pair, two chromosomes are present, right? So those are called bivalents. Bivalents, formation of bivalents, and in that bivalents together we can get four chromatids. Four chromatids will be there. Therefore, these are called tetrads. See, in a zygote stage, there is a pairing of homologous chromosomes. Pairing of homologous chromosomes, that is called synapses, and that synapses, that is due to the proteinaceous complex called Synaptonemal Complex. That all right. Synaptonemal Complex, which develops between those two homologous chromosomes, and in that paired chromosome, those are referred as bivalent cells that pair with the two chromosomes, alva? Andre paternal maternal anta heltare. Paternal maternal. Paternal from the father, paternal maternal. Paternal and maternal chromosomes pair together to form bivalent, that is synapses. That process of pairing is called synapses. And this thing, in a bivalent, 4 chromatids are present. 1, 2, 3, 4. This one chromosome with 2 chromatids, another chromosome with 2 chromatids. Together, 4 chromatids will be there, right? Say, this is first pair anta ankoli, this second pair anta ankoli. 23 pair adre 23 bivalents agate. 23 bivalents, right? And in a 23 bivalent, each bivalent with 4 chromatids. Pairing of homologous chromosomes, that is called synapses. And in a synapses, that is considered as bivalent because 2 homologous chromosomes are together, and in each pair 4 chromatids are there. Therefore, those are referred as tetrads. Tetrads. Okay. Then formation of synaptonemal complex between homologous pair. Between homologous pair, there is a formation of synaptonemal complex. Synaptonemal complex. Synaptonemal complex. Say that is a proteinaceous structure between those 2 chromosomes which ensures a pairing. Synapses ge help ಮಾಡುತ್ತೆ. Synapses ಆಗ್ಲಿಕ್ಕೆ. Say homologous chromosome ಹೇಗಿರುತ್ತೆ ಅಂದ್ರೆ structurally and genetically same. First pair, second pair. First pair ಅಂತ ಹೇಳಿದಾಗ ಆ ಒಂದು ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ ಏನೆಲ್ಲ ಜೀನ್ಸ್ ಉಂಟು ಅದೇ ಜೀನ್ಸ್ ಇದರಲ್ಲಿ ಇರುತ್ತೆ. ಅದು ಸೇಮ್ ಇನ್ ಸೆನ್ಸ್ ಇಟ್ ಮೇ ಬಿ ಅಲೀಲಿಕ್ ಫಾರ್ಮ್ಸ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಇದರ ಅಲೀಲ್ ಒಂದು gene for the body color, if it is there in a first chromosome, same gene for the body color it is there in another chromosome another. These are the pair. ಇದು ಡಾಮಿನೆಂಟ್ ಆಗಿರಬಹುದು, ಇದು ರಿಸೆಸಿವ್ ಆಗಿರಬಹುದು, ಆರ್ ಎಲ್ಸ್ ಲೈಕ್ ಇನ್ ಸಮ್ ಕಂಡೀಷನ್ ಪಾಲಿಜೆನಿಕ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅದು ಜೆನೆಟಿಕ್ಸ್ ಅಲ್ಲಿ ಬರುತ್ತೆ. ಪಾಲಿಜೆನಿಕ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಂದ್ರೆ ಅದು ತುಂಬಾ ಜೀನ್ಗಳು ಕಂಟ್ರೋಲ್ ಮಾಡುವಂತದ್ದು, ರೈಟ್? ಸೇ ದಟ್ ಮೀನ್ಸ್ ವಾಟ್ ಅಂಡ್ ಆಲ್ ಜೀನ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದಿಸ್ ಕ್ರೋಮೋಸೋಮ್, same copy will be there in another copy this particular chromosome is having, right? That pairing is a homologous chromosome. Pairing is called synapses. But this chromosome with 2 chromatids, right? ಈ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ ಇರುವಂತ DNA ಇನ್ನೊಂದು ರೆಪ್ಲಿಕೇಟ್ ಆಗಿರೋದು. ಸೊ ಒಂದು ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ 2 ಕ್ರೋಮ್ಯಾಟಿಡ್, ಇದರ ಪಾರ್ಟ್ನರ್ ಕ್ರೋಮೋಸೋಮ್ ಅಲ್ಲಿ 2 ಕ್ರೋಮ್ಯಾಟಿಡ್. ಹಾಗಾಗಿ ಬೈವೆಲೆಂಟ್ ಅಲ್ಲಿ 4 ಕ್ರೋಮ್ಯಾಟಿಡ್ಸ್ ಇರುತ್ತೆ. ಈ ಎರಡು ಹತ್ರ ಬರುತ್ತಲ್ಲ, pairing of these two chromosomes, that is synapses formation. That synapses is due to synaptonemal complex. Between 2 homologous chromosomes, there is a formation of proteinaceous structure called synaptonemal complex is present. Middle alli synaptonemal complex agutte. In that synaptonemal complex, there is a formation of recombination nodule which helps in a recombination. That is, idu zygote. Naan iga helida recombination that takes place in packaging, that will discuss later. This is about zygote stage. ಕ್ಲಿಯರ್ ಅಲ್ವಾ? ಝೈಗೋಟಿನಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಹೋಮೋಲೋಗಸ್ ಕ್ರೋಮೋಸೋಮ್ಗಳು ಹತ್ರ ಬರ್ತವೆ, ಪೇರ್ ಆಗ್ತವೆ. ಆ ಪೇರಿಂಗ್ ಅನ್ನು ಹೋಮೋಲೋಗಸ್ ಕ್ರೋಮೋಸೋಮ್ ಪೇರ್ ಆಗೋದನ್ನು ಸಿನಾಪ್ಸಿಸ್ ಅಂತ ಹೇಳ್ತೇವೆ. ಒಂದು ಸಿನಾಪ್ಸಿಸ್ ಅಲ್ಲಿ, ಒಂದು ಹೋಮೋಲೋಗಸ್ ಪೇರ್ ಅಲ್ಲಿ 2 ಕ್ರೋಮೋಸೋಮ್ ಇರುತ್ತೆ, ಹಾಗಾಗಿ ಬೈವೆಲೆಂಟ್. 4 chromatids, therefore it is tetrads. Okay, now in zygote stage, say homologous pairing, right? And that process is called synapses, right? Synapses by the help of a proteinaceous complex called synaptonemal complex. Synaptonemal complex. So if this chromatin fiber or chromosome, if it belongs to father, paternal, and this is maternal, in between these two, formation of this proteinaceous structure, right? This complex is called synaptonemal complex that helps in a formation of synapses. Synapses, pairing of homologous chromosomes called synapses, and that synapses is because of development of synaptonemal complex between these two. This is from father, this is from mother. If it is considered like that, between these two, right, there is a formation of synaptonemal complex. Between paternal and maternal chromosome, formation of synaptonemal complex leads to synapses, that is homologous pairing takes place. That is in the case of zygote. Now about packeting stage. In packeting stage, it is say first two stages, which one leptonin and zygote, that is short lived, in a sense with a short time it will complete. But zygote is very important. It is complex process and lengthy process. leptonin stage alli condensation continue ಆಗ್ತಾ ಇರುತ್ತೆ. ಕ್ರೋಮೋಸೋಮ್ಗಳು ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅಲ್ಲಿ ಕಾಣಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ, ಅಂದ್ರೆ ಕಾಣಲಿಕ್ಕೆ ಸಿಕ್ಕುತ್ತೆ. ಅದೇ zygote ಹೇಳುವಾಗ ಕ್ರೋಮೋಸೋಮ್ಗಳು ನ್ಯೂಕ್ಲಿಯೋಪ್ಲಾಸಮ್ ಅಲ್ಲಿ ಫ್ರೀ ಆಗಿರುವಂತಹ ಕ್ರೋಮೋಸೋಮ್ಗಳು ಅದರ ಪೇರ್ ಜೊತೆಗೆ ಪಾರ್ಟ್ನರ್ ಕ್ರೋಮೋಸೋಮ್ ಜೊತೆಗೆ ಪೇರ್ ಆಗುತ್ತೆ. ದಟ್ ಈಸ್ ಹೋಮೋಲೋಗಸ್ ಪೇರಿಂಗ್. ಈಗ ಪೇರ್ ಆಗಿರುವ ಕ್ರೋಮೋಸೋಮ್ಗಳ ಮಿಡಲ್ ಅಲ್ಲಿ there is another process very important, that is called crossing over. In packeting stage, important feature, that is crossing over takes place. Crossing over. What is that crossing over? Say this is from father, this is from mother. If it is considered ಒಂದು ಫಾದರ್ ಇಂದ ಒಂದು ಮದರ್ ಇಂದ ಬಂದಿರೋದು ಅಲ್ವಾ. ಒಂದು ಕ್ರೋಮೋಸೋಮ್ ಗೆ ಸೇರಿದ ಎರಡು ಕ್ರೊಮ್ಯಾಟಿಡ್ ಗಳನ್ನು ಏನು ಹೇಳ್ತೇವೆ? ಸಿಸ್ಟರ್ ಕ್ರೊಮ್ಯಾಟಿಡ್ಸ್. Chromatids of a same chromosome, sister chromatids. And if these two chromatids are considered, these are called non-sister chromatids. Chromatids of a same chromosome, sister chromatids. ಈಗ ಆ ಹೋಮೋಲೋಗಸ್ ಕ್ರೋಮೋಸೋಮ್ ಗಳು ಇರ್ತವೆ, ಪೇರ್ ಇರುತ್ತಲ್ವಾ. ಈಗ ಇದೆರಡನ್ನು ಕೇಳಿದಾಗ these two are the non-sister chromatids. Chromatids of two different chromosomes ಅಥವಾ ಆ ಹೋಮೋಲೋಗಸ್ ಪೇರ್ ಅಲ್ಲಿ ಒಂದು ಕ್ರೊಮ್ಯಾಟಿಡ್ ಒಂದು ಕ್ರೋಮೋಸೋಮ್ ಇಂದ from the father, another chromatid from the mother chromosome. These two are considered as non-sister chromatids. There is another process called crossing over takes place, that is between two non-sister chromatids. That process means exchange of genetic material between two non-sister chromatids. ಇದೆರಡರ ಮಿಡಲ್ ಆಗೋದಿಲ್ಲ, ಈ ಹೋಮೋಲೋಗಸ್ ಪೇರ್ ಅಲ್ಲೇ ಆಗುತ್ತೆ. Exchange of genetic material between two non-sister chromatids called, that is that process is called crossing over. Exchange of genetic material, exchange of genetic material between two non-sister chromatids, that is called crossing over. What will happen? Exchange of chromatids takes place. At that particular point where crossing over takes place, that region is called chiasmata. Chiasmata is a region where crossing over takes place. Okay, ಎರಡು ಕ್ರೊಮ್ಯಾಟಿಡ್ ಗಳು ಅಂದ್ರೆ ಇಲ್ಲಿ ಒಂದರ ಮೇಲೆ ಒಂದು ಬಂದಾಗ ಈ ಪರ್ಟಿಕ್ಯುಲರ್ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ ಜೀನ್ ಎಕ್ಸ್ಚೇಂಜ್ ಆಗುತ್ತೆ. ಈ ಕ್ರೋಮೋಸೋಮ್ ಇಂದ ಇದಕ್ಕೆ, ಇದರಿಂದ ಇದಕ್ಕೆ genetic exchange takes place, and that is that process that leads to recombination. It leads to recombination of genes. This recombination, that is with the help of an enzyme called recombinase enzyme. Here in the case of packeting stage, paired chromosome, homologous pair, which will be easily observed, clear ಆಗಿ ಕಾಣುವಂತದ್ದು. zygote-ನಲ್ಲಿ ನಮಗೆ bivalent ಅಂತ ಹೇಳಿದ್ರು ಕೂಡ ಪ್ಯಾಕೆಟಿಂಗ್ ಸ್ಟೇಜ್ ಅಲ್ಲಿ ತುಂಬಾ ಕ್ಲಿಯರ್ ಆಗಿ ಕಾಣುತ್ತೆ, bivalent with the tetrad condition. At that particular point, with the help of synaptonemal complex, already synapses ಆಗಿದೆ, pair ಆಗಿದೆ, exchange of genetic material takes place, right? Exchange of genetic material between two non-sister chromatids. ಇದೆರಡಲ್ಲ, ಯಾಕೆ ಇದ್ರದ್ದೇ ಕಾಪಿ ಇರೋದಲ್ವಾ, ಇಲ್ಲಿಯ ಮತ್ತೆ ಇಲ್ಲಿಯ ಮಿಡಲ್ ಅಲ್ಲಿ ಆಗುವಂತದ್ದು ಎಕ್ಸ್ಚೇಂಜ್ ಆಫ್ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಬಿಟ್ವೀನ್ ಟು ನಾನ್ ಸಿಸ್ಟರ್ ಕ್ರೊಮ್ಯಾಟಿಡ್ಸ್. Therefore that process is called crossing over, and crossing over that takes place in a point called that recombination nodule. ಹೇಗಾಗುತ್ತೆ ಅಂದ್ರೆ ಈ ಕ್ರೊಮ್ಯಾಟಿನ್ ಫೈಬರ್ ಇಲ್ಲಿ ಬರುತ್ತೆ, ಈ ಕಡೆಯಿಂದ ಇದು ಇಲ್ಲಿ ಬರುತ್ತೆ. ಈ ಪರ್ಟಿಕ್ಯುಲರ್ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ ಇದನ್ನು ನಾವು ರೀಕಾಂಬಿನೇಷನ್ ನೊಡ್ಯೂಲ್ ಅಂತ ಹೇಳ್ತೇವೆ. ರೀಕಾಂಬಿನೇಷನ್ nodule formation. Recombination nodule formation takes place. This chromatin it moves like this, and this chromatin it moves like this. Between these two chromatin fibers of paternal and maternal, which exchanges gene exchange ಆಗುವುದು. ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಆದಾಗ ಇದರ ಕ್ರೊಮ್ಯಾಟಿನ್ ಫೈಬರ್ ಜೊತೆಗೆ ಜೀನ್ ಎಕ್ಸ್ಚೇಂಜ್ ಮಾಡ್ಕೊಳ್ಳುತ್ತೆ. ಆ ಪಾಯಿಂಟ್ ಅನ್ನು ನಾವು ಕಯಾಸ್ಮಟ ಅಂತ ಹೇಳ್ತೇವೆ. ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಆಗುವ ರೀಜನ್ ಅದು ಕಯಾಸ್ಮಟ. Region where crossing over takes place, that point is chiasmata, and that recombination is with the help of enzyme called a recombinase enzyme. ಆ recombine ಆಗುವಂತ ಪಾಯಿಂಟ್ ಇರುತ್ತಲ್ಲ ಅದನ್ನ ನಾವು recombinate nodule ಅಂತ ಹೇಳ್ತೇವೆ. recombination nodule ಅಂತ ಕರೀತೇವೆ. Say this is about packaging. I'll write the diagram. ಇದನ್ನು ಬರ್ಕೊಳ್ಳಿ. What is crossing over? That exchange of genetic material between two non-sister chromatids. That point where crossing over takes place, that region is chiasmata with the help of recombinase enzyme, and at that point recombination nodules are formed. This is the identification point for the packaging stage. Okay. About the packaging, as I said now, that homologous pairing and crossing over takes place. Here you can make out this homologous pairing, exchange of genetic material between two homologous chromosomes. Exchange of genetic material between two non-sister chromatids. Two non-sister chromatids in the ಬರೆಯೋಬದಲಿಗೆ between two homologous chromosomes, right? ಈಗ homologous chromosome ಅಲ್ವಾ, ಇದರ partner chromosome, right? So genes which exchanges at that particular point, this point is called chiasmata, where crossing over takes place. Exchange of genetic material between two non-sister chromatids or homologous chromosomes. This is about the packaging stage. Here, as I said now, say one chromatin fiber and the chromosome paternal, ಇನ್ನೊಂದು maternal ಅಲ್ವಾ. ಇಲ್ಲಿ synaptonemal complex ಇಂದಾಗಿ ಪೇರ್ ಆಗಿದೆ. ಆ ಪೇರ್ ಆಗೋ ಪರ್ಟಿಕ್ಯುಲರ್ ರೀಜನ್ ಅಲ್ಲಿ in particular points where recombination nodule formation ಆಗುತ್ತೆ. ಆ nodule formation ಆದ region ಅಲ್ಲಿ genetic material exchange ಆಗುತ್ತೆ. Loop formation, chromatin fiber, it forms a loop and that particular nodule region where exchange of genetic material takes place. Okay, this is the end of packaging stage. At the end of packaging stage, crossing over positions will be there, chiasmata region will be there, where genetic material exchanges between two non-sister chromatids of homologous chromosomal pair. This is about the packaging. Next one is diplotin and diakinesis. So ಇಲ್ಲಿ ಗೊತ್ತಾಗುತ್ತಲ್ಲ, ಇದು ಪೇರ್ ಆದದ್ದು, ಇಲ್ಲಿ ಕ್ರೋಮೋಸೋಮ್ಗಳು ಎಕ್ಸ್ಚೇಂಜ್ ಮಾಡ್ಕೊಳ್ತಾರೆ. ಈ ಜೀನ್ಗಳ ಅಂದ್ರೆ ಇಲ್ಲಿ ಕ್ರೊಮ್ಯಾಟಿಡ್ಗಳು ಎಕ್ಸ್ಚೇಂಜ್ ಮಾಡ್ಕೊಳ್ಳೋದಂದ್ರೆ ಪರ್ಟಿಕ್ಯುಲರ್ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ ಜೀನ್ ಎಕ್ಸ್ಚೇಂಜ್ ಆಗುತ್ತೆ. ಆ ಪರ್ಟಿಕ್ಯುಲರ್ ಪಾಯಿಂಟ್ ವೇರ್ ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್, ದಿಸ್ ಎಕ್ಸ್ಚೇಂಜ್ ಸ್ಟ್ರಕ್ಚರ್ ದಟ್ ಈಸ್ chiasmata. That depends upon the length of the chromosome. ಎಲ್ಲೋ ಎಕ್ಸ್ಚೇಂಜ್ ಆಗೋದು ಈ ಕ್ರೋಮೋಸೋಮಲ್ ಪೇರ್ ನ ಮಿಡ್ಲ್ ಅಲ್ಲಿ. ಇದು ಪ್ಯಾಕೆಟಿಂಗ್ ಸ್ಟೇಜ್ ಅದು. ಈಗ ಡಿಪ್ಲೋಟಿನ್ ಮತ್ತೆ ಡಯಾಕೈನೆಸಿಸ್ ನೋಡುವ. Diplotin stage, after the packaging, diplotin stage, and at diplotin stage, that synaptonemal complex which is formed between two homologous chromosomes, it starts to dissolve, or dissolution of synaptonemal complex. Synaptonemal complex between synaptonemal complex between homologous chromosomes. Homologous chromosomes. And thereby say synaptonemal complex form ಆದಕ್ಕೆ synapses ಆಯ್ತು, homologous pair ಆಯ್ತು ಅಲ್ವಾ. ಎರಡು ಕ್ರೋಮೋಸೋಮ್ ನ ಮಿಡ್ಲ್ ಅಲ್ಲಿ ಆ synaptonemal complex ಫಾರ್ಮ್ ಆದದ್ದು. Now dissolution of synaptonemal complex between these two chromosomes, by that these chromosomes starts to move away from each other except in a region called chiasmata where crossing over is there. Okay, dissolution of synaptonemal complex between homologous chromosomes except the region of crossing over. Except the region of crossing over, that is nothing but chiasmata. Region of crossing over are the region bittu. Crossing over, that is chiasmata. Chiasmata region bittu ulda region alli a synaptonemal complex dissolve aguttalla. By that chromosomes from the pair, ಈಗ pair ಆಗಿದಲ್ವಾ, ಆ pair ಇಂದ chromosome ಗಳು separate ಆಗಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡುತ್ತೆ. Say that is the end, ಅಂದ್ರೆ diakinesis, that diplotin ಆಯ್ತು. Now about the diakinesis, diplotin ಇದಾಯ್ತಲ್ವಾ, diakinesis. In diakinesis stage, thick and condensed chromosome with the exchange genes, with the exchange genetic material. ಅಂದ್ರೆ ಈಗ ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಆಗಿರುವಂತದ್ದು, there is a terminalization of crossing over ಅಂತ ಹೇಳ್ತೇವೆ. ಎಂಡ್ ಆಗ್ತಾ ಬಂದ್ರೆ ಆ ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಬಿಟ್ರೆ ಮತ್ತೆ ಉಳಿದ ಎಲ್ಲಾ ಪೊಸಿಷನ್ ಇಂದ ಕ್ರೋಮೋಸೋಮ್ಗಳು ದೂರ ಆಗಿವೆ. From that chromosomal pair, right, except the crossing over region, that chromosome is separated from that complex. Synaptonemal complex dissolve ಆಗೋದ್ರಿಂದಾಗಿ ಕ್ರೋಮೋಸೋಮ್ಗಳು ದೂರ ಬಂದಿದ್ದಾರೆ. ಓಕೆ, ಆ ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ ಮಾತ್ರ ಜಾಯಿನ್ ಆಗಿರುವಂತದ್ದು. ಈಗ ಆ ಟರ್ಮಿನಲೈಸೇಷನ್ ಅಂದ್ರೆ ಎಂಡ್ ಆಗ್ತಾ ಬಂತು, ಎಕ್ಸ್ಚೇಂಜ್ ಆಫ್ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್. Now chromosomes which are thick and short condensed chromosomes with the exchange genetic material is present. Now it is that phase which was diakinesis, the transition to metaphase, and that particular stage nuclear membrane disappears, nucleolus disappears, all those events takes place in a diakinesis. That is a transition zone or transition phase, transition to metaphase first. Okay, this is about the prophase first, which is the lengthiest process and complex process. Meiosis na ಏನೆಲ್ಲ ಸಿಗ್ನಿಫಿಕೆನ್ಸ್ ಹೇಳ್ತೇವೆ ಅದರಲ್ಲಿ ಮೇನ್ ರೀಸನ್ ಯಾವುದು, ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಮತ್ತೆ ರಿಕಾಂಬಿನೇಷನ್ ಆಗುವಂತದ್ದು prophase first ಅಲ್ಲಿ. Leptotin stage where chromatin materials which starts to condense or condensation of chromatin materials takes place. Chromosomes are visible at that particular point, that is leptotin. Zygote where homologous chromosomes starts to pairing of homologous chromosomes takes place, that is called synapses. And that synapses is because of the complex, proteinaceous complex formation between those two chromosomes, that is synaptonemal complex. Okay, these two stages, leptotin and zygote stages, are short-lived. Next one is packeting. In a packeting stage, there is a crossing over takes place. Very important, crossing over, exchange of genetic material between two non-sister chromatids of homologous pair. ಆ homologous pair ನ ಒಳಗೆ non-sister chromatid ನ ಮಿಡ್ಲಲ್ಲಿ gene exchange ಆಗುತ್ತೆ. Okay, that is in a packeting stage, and that is with the help of enzyme called recombinase enzyme. And one more thing is formation of recombination nodules. ಆ particular point ಅಲ್ಲಿ recombination nodule formation ಆಗುತ್ತೆ. ಆ ಪಾಯಿಂಟ್ ಅಲ್ಲಿ gene exchange ಆಗೋದು. ಈಗ ಕ್ರೋಮೋಸೋಮ್ homologous pair ಅನ್ನು bivalent ಅಂತ ಹೇಳ್ತೇವೆ, ಟೆಟ್ರಾಡ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಯಾಕೆ? 4 chromatids are there. Packetine stage ಅಲ್ಲಿ crossing over ಆಯಿತು. After the packetine diplotin stage ಅಲ್ಲಿ ವಾಪಸ್ ಸಿನಾಪ್ಟೋನಿಮಲ್ ಕಾಂಪ್ಲೆಕ್ಸ್ ಫಾರ್ಮ್ ಆದದ್ದು ಡಿಸಾಲ್ವ್ ಆಗಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಡಿಸಾಲ್ವ್ ಆದಾಗ ಕ್ರೋಮೋಸೋಮ್ಗಳು ದೂರ ಹೋಗ್ಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಎಲ್ಲಿ ಕಯಾಸ್ಮಟ ರೀಜನ್ ಬಿಟ್ಟು. ಓಕೆ, ನಾವು ಅಬೌಟ್ ದ ಡಯಾಕೈನೆಸಿಸ್ ಕಂಪ್ಲೀಟ್ ಟರ್ಮಿನಲೈಸೇಷನ್ ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಆದದೆಲ್ಲವೂ ಈಗ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಆಲ್ರೆಡಿ ದೂರ ದೂರ ಬಂತು. Now the terminalization of that crossing over at that particular point, that is diakinesis, nuclear envelope, nucleolus, all those organelles that starts to disappear. Disintegration of nuclear envelope takes place, and that is the transition towards metaphase. Next, we'll study about the metaphase first.